Everyone, I am Naveen Shreevastva. I have been working as a biochemist at Agriculture and Forestry University, Chitwan, Nepal. So today I am going to talk about the replication process. So in replication process, we are going to talk about the DNA replication. So we know that the process of replication forms replica of DNA, that is, it forms the copy of DNA. So one of the major points that we have to remember is that when DNA undergoes replication, one parent DNA forms two daughter DNA. So by the process of replication, we are going to get two daughter DNA from one parent DNA. Another important point is that this process of replication is called semi-conservative replication. It is called semi-conservative replication. So now we have to know why is it called semi-conservative replication. It is called semi-conservative replication because when two daughter DNAs are formed from one parental DNA, half of the character of parent DNA is present in one of the daughter DNA and another half of the parental DNA character is present in another daughter DNA. So we can explain it by the help of a figure. So suppose this is one of the parental DNA, and this parental DNA undergoes replication process. So when this parental DNA undergoes replication process, it will generate two daughter DNA. So what happens that this is the polarity of the strands. So when two daughter DNAs are formed from one parental DNA, one of the strands of parental DNA is in one daughter DNA and another parental strand is present in another daughter DNA. So suppose this is this 5' to 3' strand is present in this daughter DNA. So this daughter DNA will get one parental strand from parent and another strand is the newly formed strand. So this another strand will be 3' to 5'. So finally we get one daughter DNA in which one parental DNA strand is there and another newly formed strand is there. At the same time, in another daughter DNA, what happens that this is 3' to 5' strand, then another newly formed strand moves from 5' to 3' because we know that the two strands of DNA are anti-parallel in nature. So finally we are at the point why is it called semi-conservative replication. So the figure itself explains that in both daughter DNA, one of the parent DNA strand is there, another strand is newly formed, and in other DNA also one of the there is one parental DNA strand and another strand is newly formed strand. So since half of the DNA strand is in one daughter DNA and another half is present in other daughter DNA, that's why it is called that's why this replication process is called semi-conservative replication because due to the partial conservation of character of parental DNA into daughter DNAs. So this is a major point. Now we have to know what is the process of replication in prokaryotes and what is the process of replication in eukaryotes. So we know that in prokaryotes and in eukaryotes, the basic steps are similar, but the involvement of compounds are totally different. So at first we are going to talk about the DNA replication process in prokaryotes, then we talk about the DNA replication in eukaryotes. So first coming to DNA replication in prokaryotes. So DNA replication in prokaryotes, we know that prokaryotes are bacteria or prokaryotes. So simply we are going to talk about the replication process in bacteria finally. So before going through the different steps of DNA replication in prokaryotes, at first we have to know what are the different requirements for the DNA replication process. So at first we will talk about the requirements. So since we have to form daughter DNA molecules from parental DNA, so one of the major requirements is deoxyribonucleotides. Deoxyribonucleotides, we know that 4 different types of deoxyribonucleotides are needed for the formation of daughter DNA, and they are DATP, DCTP, DGTP and DTTP. That is deoxyadenosine triphosphate, deoxycytidine triphosphate, deoxyguanosine triphosphate and deoxythymidine triphosphate. Always we have to remember one point is that inside the structure of DNA, these deoxyribonucleotides are in monophosphate form. That is, this DATP is inserted in the structure of DNA in the form of DAMP. It is inserted in the form of DAMP, but as a requirement it is DATP. Then this CTP is inserted in the form of DCMP, but required in the form of DCTP. It is inserted, DGTP is inserted in the form of DGMP, and DTTP is inserted in the form of DTMP. That is, all the deoxyribonucleotides are inserted in the form of monophosphate, but they are required in the form of triphosphate. So we have to know why this type of triphosphate molecules are needed as a raw material. So we know that when there is formation of DNA, the deoxyribonucleotides are held together by the help of phosphodiester bond. We know that. Suppose this is DNA strand, and suppose this A stands for suppose DAMP and this G stands for DGMP. So we know that in an individual strand of DNA there is presence of phosphodiester bond between this DAMP and DGMP. So always remember, this triphosphate form is more active than this monophosphate form. So since we have to make, or since the bacterial body has to make a phosphodiester bond between 2 deoxyribonucleotides, so these molecules are needed in the form of triphosphate. If they are not present in triphosphate form, phosphodiester bond is not going to be formed, and finally DNA strand cannot be formed. So one of the requirements is deoxyribonucleotides in the form of triphosphate. Then another protein is required, that is called DNA A protein. We will discuss the role of these proteins later on. Then DNA A protein, then there is need of helicase enzyme, DNA helicase. It is called DNA helicase, or it is simply called as DNA B protein. DNA B protein. So this is DNA B protein. Then there is need of single strand binding protein. Single strand binding protein, which is simply called as SSBP. It is simply called SSBP, single strand binding protein. Then there is need of DNA polymerase 3, DNA polymerase 3, then DNA polymerase 1, DNA polymerase 1, then there is need of primates, primase enzyme, then there is need of DNA ligase, and last but not least, topoisomerase, that is called DNA topoisomerase. DNA topoisomerase is required. So by using these type of requirements, we can simply, or the bacterial body can simply synthesize daughter DNA from parental DNA molecules. So after the requirements, we are going to talk about the different steps of DNA replication. So we broadly divide the steps of DNA replication into 3 different types. One, initiation of DNA replication, that is simply called as initiation process. So this is the actual initiation of DNA replication. Then elongation of new DNA strand, and third one is termination process. So we will be discussing all these steps separately, one by one.